Hi, this is KKR sir, Biology NEET faculty. Now we are explaining about the vector subtopic insertional inactivation. Insertion of DNA fragment at one position, that site, the position lost their capacity is called insertional inactivation. See, for example, here, very few or only one cloning site is required for multiplication of desirable gene, earlier we told. For example, in PBR 322, BAM-H1 is a cloning site. It has two antibiotic resistant genes. One is ampicillin resistance, another one tetracycline resistance. The desirable gene is a linking of BAM-H1 site at tetracycline resistance capacity. When desirable gene is linked to the tetracycline resistance, the inactivation of tetracycline resistance is called insertional inactivation. Mean one side tetracycline, one side ampicillin resistance there. When desirable gene is linked to the tetracycline resistance, the inactivation of tetracycline resistance loss is insertional inactivation. Then how it is, sir, useful? However, it lost tetracycline resistance capacity, we can identify the recombinants by transforming to the ampicillin plate. Means these transformants with the recombinant DNA, when transformed to the ampicillin plate, they can grow nicely. They cannot grow in a tetracycline plate because they lost tetracycline resistance. See here, three words I will give. Non-transformants with the normal bacteria. Normal bacteria, no transformant, nothing. Not having any resistance to the ampicillin and a tetracycline. So they cannot grow in an ampicillin plate and they cannot grow in a tetracycline plate. Transformants with the recombinant DNA, they can grow in an ampicillin plate only, not grow in a tetracycline plate, because they lost a tetracycline resistance capacity and they had only ampicillin resistance capacity. Bacterial cell with artificial plasmid, no insertion, they can grow in ampicillin, they can grow in a tetracycline also, because both they had antibiotic resistance capacity. This word understand, try to understand. Then this seems to be some cumbersome process. That's why identification of recombinant DNA or transformants, there is an alternate method had been developed, because two plates of antibiotic resistance, these are all some clumsy nature is there. Simple. Producing color based on the chromogenic substrate on reporter enzyme. A concept देखो, for example, bacterial cell has beta galactosidase enzyme. Due to the presence of beta galactosidase enzyme, they can grow and then producing blue color colonies due to the beta galactosidase enzyme. Of course, when insertion is there, when insertion at a beta galactosidase, the beta galactosidase is inactivated due to insertional inactivation. Normal bacteria cells produce blue color colonies due to the presence of beta galactosidase enzyme. When insertion, recombinants or transformants producing no color colonies due to the inactivation of beta galactosidase. Blue color colonies, non-transformants. No color colonies or white color. No color first, other white color. Colonies, transformants or recombinants. This is nothing but insertional inactivation. Okay, thank you.